Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, at the very heart of life, there's this profound instruction manual, a code that dictates every single thing about us. But this manual isn't just meant to be read. It has to be copied, perfectly, billions and billions of times. So today we're going to dive into the absolutely incredible process of DNA replication.
Take a look at this number, 6 billion. That's a mind-boggling number, right? Well, that's how many base pairs, the individual letters of our genetic code, are packed into just one of your diploid cells. And every single time that cell divides, this entire library has to be copied with almost perfect accuracy. So let's explore the amazing molecular choreography that makes this incredible feat possible.
So the official term for this whole process is DNA replication. And the goal really is simple in concept, but absolutely staggering when you think about the execution. It's all about making sure that each new daughter cell gets a completely identical copy of the original genetic blueprint. This continuity is the very foundation of heredity and of life itself.
But here's the really cool part. It's how the copy gets made. It's not like the cell builds a new DNA molecule from scratch. Instead, it uses what we call a semi-conservative model. What this means is that every new DNA double helix is actually a hybrid. One strand is the original, the one from the parent, and the other is brand new, built using that original strand as a perfect template.
And you know, this wasn't just some wild guess. It was proven back in 1958 by a landmark experiment from Messelson and Stahl. They cleverly tagged bacterial DNA with a heavy version of nitrogen. After one round of replication in a light medium, they didn't find heavy DNA or light DNA. They found a single band of hybrid DNA right in the middle. It was proof that each new molecule was half old and half new, which totally cemented the semi-conservative model as a fundamental principle of biology.
So we know how it happens, in principle, but every story has to have a beginning. With 6 billion letters to copy, where on earth does this monumental task actually start?
Think about it. The DNA double helix. It's famously stable. I mean, that's fantastic for keeping our genetic information safe, but it presents a real challenge for replication. Before you can copy it, you have to pry those two strands apart. The cell has to be incredibly precise about where this unwinding process begins.
Well, it all kicks off at these specific spots called origins of replication, or ORIs for short. A special group of proteins called the origin recognition complex, the ORC, acts like a scout finding these starting points. Once it's found a spot, an enzyme called helicase gets to work, unzipping the two strands of the helix, just like a zipper on a jacket. And to stop the strands from immediately snapping back together, these little single-strand binding proteins come in and coat the exposed DNA, holding it open and ready for the copying to begin.
All right, so the DNA is open. Now it's time to call in the main construction crew. And this isn't just one enzyme doing all the work. It's a complex molecular machine called the replisome.
And here's a look at that machine in action, at what we call the replication fork. That's the point where the DNA is actively splitting. I know there's a lot going on here, but just imagine a perfectly coordinated team. You've got helicase at the very front unwinding the DNA. Right behind it, primase lays down a little starting block. And then comes the star of the show, DNA polymerase, which actually builds the new strand. It's this beautiful coordinated dance of proteins working together.
But there's a major roadblock, a big logistical puzzle that this whole machine has to solve. The two strands of the DNA helix are anti-parallel. They run in opposite directions, kind of like a two-way street. The problem is, DNA polymerase can only build in one direction. So how does the cell copy both strands at the same time when they're pointing in opposite ways?
The solution is just incredibly clever. It's so elegant. The cell simply synthesizes the two new strands differently. One strand, which we call the leading strand, is built in one smooth continuous piece because it's already pointing in the right direction. But the other strand, the lagging strand, is synthesized discontinuously in these small chunks called Okazaki fragments, which are then stitched together later.
So let's just quickly walk through how this works for that tricky lagging strand. First, an enzyme called primase lays down a short little RNA primer to give the polymerase a place to start. Then, DNA polymerase comes in and synthesizes a short Okazaki fragment, building backwards away from the replication fork. After that, another type of polymerase comes in, swaps out that RNA primer for actual DNA, and finally, an enzyme called DNA ligase acts like molecular glue, joining all the fragments together into one complete, continuous strand. It's an amazing process.
Okay, so the copy is made, but think about it. We're copying 6 billion letters. Mistakes are bound to happen, right? And a single error can have devastating consequences. So the cell has this extensive quality control system, a whole suite of DNA repair mechanisms.
And this is the bottom line, right here. Fidelity is everything. The copying has to be as accurate as possible. If the genetic information gets altered, the consequences can be huge, from a cell not working right all the way to serious diseases. The cell just can't afford to be sloppy, so it has evolved these robust, pretty much foolproof ways to find and fix mistakes.
And it's not just one fix-it tool. The cell has this whole arsenal of repair systems for different kinds of damage. Mismatch repair is there to fix simple typos made during copying. Then you have nucleotide and base excision repair, which handle chemical damage from things in our environment. And what's absolutely incredible is that there are even mechanisms to fix catastrophic double-strand breaks, the kind caused by things like radiation.
The very fact that these repair systems exist tells you just how vital they are. But what happens when one of these systems fails? When it's broken? Well, the consequences can be seen in a number of really serious human diseases.
A really tragic example of what happens when things go wrong is a condition called xeroderma pigmentosum. In people with this condition, the nucleotide excision repair system, that's the one that's supposed to fix damage from the UV light and sunshine, is broken. Because of this, they are extremely sensitive to sunlight, and even just a little bit of exposure can lead to a massively increased risk of developing skin cancer.
So this is the molecular assembly line that's broken in these patients. Normally, a set of proteins would spot the UV damage, enzymes would come in and snip out the wrong section of DNA, and then others would synthesize the correct patch to fill the gap. But in xeroderma pigmentosum, a critical part of this assembly line is missing or defective. The damage from UV light just builds up, and that leads directly to the devastating symptoms of the disease. It's such a powerful, direct link between a single molecular pathway and a human disease.
And that brings us to this final, really profound question. We've just seen this incredibly sophisticated, beautiful system of replication and repair, a process that's been fine-tuned over billions of years of evolution. So, if our cells are this good at maintaining their genetic code, why aren't they perfect? Why do these systems eventually start to falter, leading to things like aging and diseases like cancer? It's a humbling reminder, isn't it, that even the most elegant biological systems have their limits.